technique of plant tissue culture. Plant tissue culture is a technique of growing plant cell tissues, organ seeds, or other plant parts in a sterile environment on a nutrient medium. Basic techniques of plant tissue culture: culture vessels, culture medium, serialization, inoculation, incubation of callus, morphogenesis or is equals to organogenesis and plus embryogenesis. Hardening. Cultures can be grown up in various kinds of vessels such as petri plates, test tubes, magenta boxes, bottles, and flasks. Culture medium, the important media used for all purpose experiment are murashich and hooch medium, MS medium. The culture medium is closed with cotton plug or aluminum foil sheet. The pH of the medium is adjusted to 5.8 acidic range. Composition of culture medium. Nutrient medium depends upon the type of plant tissue or cell used for culture. Generally, nutrient consist of inorganic salts. Both micro and macro elements are carbon source, usually sucrose, vitamins, nicotinic acid, thiamine, pyridoxine, amino acids, arginine, growth regulators, auxins, and optimum pH 5.7 is also very important. Serialization. Serialization methods used in tissue culture laboratory. All the materials, for example, vessels, instruments, medium, plant material, etc., used in culture work must be freed from microbes. Sterilization techniques. Sterilization is achieved by one of the following approaches: dry heat treatment, flame sterilization, autoclaving, filter sterilization, wiping with 70% alcohol, surface sterilization. Inoculation. Transfer of explant, root, stem, leaf, etc. onto a culture medium is called inoculation. The inoculation is carried out under aseptic conditions for which an apparatus called laminar flow chamber is used. Flamed and cooled forceps are used for transfer of plant material to different culture media kept in glasswares. Incubation. The culture medium with the inoculum is incubated at 26 to 28 degrees centigrade with the light intensity at 2000 to 4000 lux, unit of intensity of light, and allowing allowing photo period of 16 hours of light and 8 hours of darkness. Induction of callus. Due to activity of auxins and cytokinins, the plant is induced to form callus. The callus is an unorganized mass of undifferentiated tissue. The mechanism of callus formation is that auxin-induced cell elongation and cytokinin induces cell division, as a result of which masses of cells are formed. Morphogenesis. Morphogenesis, formation of new organs from the callus under the influence of auxin and cytokinin, is called morphogenesis. Roots and shoots are differentiated from the callus. Such embryos are called somatic embryos, result in the formation of young plantlet. Types of morphogenesis: organogenesis, embryogenesis. Organogenesis, formation of new organs such as shoot and root, is known as organogenesis. The development of shoot from the callus is called collagenesis, and formation of root is called rhizogenesis respectively. Embryogenesis, formation of embryos, bipolar structure having shoot and root from the callus is called embryogenesis. These embryos arise from somatic callus tissue and are called somatic embryos or embryoids or soma clonal embryos. Hardening. Exposing the plantlets to the natural environment in a stepwise manner is known as hardening. Finally, the plantlets are gradually transferred to the soil. Types of tissue culture, callus culture. Types of tissue culture. Types of in vitro cultures. Callus cultures, cell suspension cultures, anther or microspore culture, protoplast culture, embryo culture, meristem culture. Types of in vitro culture: plant tissue culture, seed culture, bud culture, meristem culture, protoplast culture, organ culture, embryo culture, cell culture, suspension culture, callus culture. Callus culture. An unorganized mass of thin-walled parenchyma cells formation of bone callus is observed in all groups of living organisms in plant tissues. In plant tissue culture, callus is produced by cultivation of plant tissue on nutrient media under in vitro conditions. Presence of growth hormones in the culture media promotes promotes callus formation and proliferation. Role of callus: in vitro callus provides totipotent cells for plant regeneration via organogenesis or somatic embryogenesis. Callus is used as a target tissue for genetic transformation. Callus formation is initiated for plant regeneration of other transformed tissues. Dispersal of friable callus into single cells is used for the initiating cell suspension cultures. Transgenic callus induction, explant callus induction, transgenic callus transformation, callus and suspension cultures, transgenic callus organogenesis, and callus somatic embryogenesis, organogenesis, somatic embryogenesis, and suspension culture, somatic embryogenesis, and plantlet and transgenic plantlet. Initiation and maintenance of callus culture, selection of suitable parent material, choice of explant and method of isolation, culture medium and conditions required, optimization of culture conditions, selection of suitable parent plant must be healthy and free from decay or disease. Mother plant be actively growing and not be about to enter a period of dormancy. Choice of plant explant and method of isolation: any part of mother plant organs or specific plant tissue or plant cells. Explant must contain living cells. Younger tissue is more callus responsive due to the presence of large number of actively dividing cells. Explants isolated in sterile conditions ensure proper sterilization method for a particular explant. Culture medium and conditions required. Culture culture the sterilized explants on suitable autoclaved culture medium. Incubate cultures at 22 to 24 plus minus 2 degrees centigrade in light or dark. Most callus will be initiated from the cut surfaces within 3 to 8 weeks. Optimization of culture conditions. Consult literature to know previous callus initiation attempts for species under consideration. For pioneering callus culture attempt, modify medium previously used for a related species. Start with one of the defined media and manipulate hormone concentrations. Set up a growth Latin square of 25 culture plates with 5 each of auxin and cytokinin concentration. Callus for this growth should be uniform of large 20 mg size. Callus growth measurement subculture vigorous growing callus 2 to 5 millimeters diameter. Slow growing callus plus explant transfer to fresh medium. Callus growth assessment via fresh and dry beds. Plot a growth curve. Thank you.